Hello guys, so let's talk about genetic expression. In this case, we'll talk about DNA and transcription and translation into proteins. So DNA, the process of DNA that happens in the nucleus of the cell is transcription. So the messenger RNA is going to make a copy of DNA, and then the messenger is able to go outside of the nucleus into the cytoplasm, and then the next process is going to happen. This process is translation. So messenger is transformed or translated into proteins. So we're going to see what happens. Let's do a review about this. So remember, the RNA has three types: messenger, uh, ribosomal, and transfer RNA. Then the RNA is called ribonucleic acid, and then we need for this process one strand of nucleotides from DNA. We need the sugar that is going to be different. In this case, it's going to be ribose. Ribose instead of being deoxyribose, and the uracil instead of thymine. That will be another change. So the messenger RNA, we have messenger, ribosomal, and transfer. The messenger RNA will transcript the genetic information from the synthesis of proteins from DNA towards to the cytoplasm, and the RNA has a sequence of bases, nitrogenous bases, that acts as a pattern to make amino acids on the protein to be synthesized. So the rRNA or ribosomal RNA, this molecule along with the ribosomal protein build the big and small subunits that constitute ribosomes. The ribosomes link to the messenger RNA molecule to translate to a sequence of amino acids, the information contained in the sequence of nucleotides present in the messenger RNA. Then the transfer or tRNA. It is a small molecule that transports amino acids from the cytoplasm towards to the ribosomes, where they are organized according to the information from messenger RNA. So therefore, together with ribosome, the transfer is responsible for translating the genetic code. So the transcription and translation. Let's start with transcription. So DNA, the sequence of nucleotides determine the sequence of amino acids of proteins. The information that DNA contains is not used direct from DNA. So the RNA acts as an intermediary between DNA and proteins. The first step for this is the transcription. So transcription from DNA into messenger RNA. So in this way, the DNA won't go outside of the nucleus of the cell, and that will stay there. So before the transcription takes place, the DNA double helix must be unwound, because in this case, we're going to use just one of the strands of DNA. And for that, we need to have this transcription bubble. So here we have in this, the transcription use one of the two exposed strands of DNA, and this is called the template strand, while the other one is called the coding strand. The template strand has the messenger RNA. The messenger RNA is a complement of the template strand, and the non-codifying strand is also called non-template strand. Remember that RNA doesn't have thymine, but instead of that, it has uracil. And the other thing, the sugar won't be the deoxyribose, but the ribose. So to start transcription, we need a site called initiation site or +1 site. In eukaryotic cells, there will be extra process between transcription and translation. But what we need here is the RNA pol that will bind to DNA and at one place called promoter. Basically, the promoter will tell the polymerase where to sit down on DNA that is being transcribed. So the transcription initiation. For the transcription initiation, DNA transcribing a gene. The RNA pol binds to the DNA of the gene and region called promoter. Basically, the promoter tells the polymerase where to sit on DNA and be transcribing this. Each gene has its own promoter, and once the transcription bubble has formed, the polymerase can start transcribing. The promoter contains DNA sequence that let RNA pol or its helper attach to DNA. In the case of eukaryotic cells, promoters in humans, um, the main RNA pol in your cell does not attach directly to the promoter. So, not directly to the promoter right here. Instead of that, there are helpers or called basal general transcription factors, basal or transcription factors. They will help the RNA pol sets get a foothold on DNA and let know where it's supposed to go. So many eukaryotic promoters have a sequence called data box, and this data box is recognized by one of the general transcription factors, allowing other transcription factors identify in this place, and eventually the RNA pol will bind to this structure, which make it easy to pull the strands of DNA apart. Elongation, the next step. Elongation, as you see here, RNA is growing. So the direction of the chain is 3' to 5'. During the elongation, the RNA polymerase will walk along one strand of DNA known as the template strand in 3' to 5' direction. For each nucleotide in the template, RNA pol adds a matching complementary RNA nucleotide to the 3' end. The RNA transcript is nearly identical to the non-template or coding strand of DNA. However, RNA strands have a base of uracil in place of thymine, as well as slightly different sugar in the nucleotide. So instead of deoxyribose, it has ribose. So the last part is termination. So here we have, okay, in different cells, there won't be, like bacteria, there won't be this extra genetic material. So in termination, the RNA will be mature. But in the case of eukaryotic cells, there is one more step before the messenger RNA is ready to be used. So the RNA, in this case, is a pre-messenger RNA with extra material, the purple or introns. The introns are extra genetic material and it needs to be eliminated. And the cell is going to use three processes to eliminate this. One process is splicing, then other one is editing, and the last one is polyadenylation. So in splicing, what happens? Splicing removes the introns of the RNA molecule. And the messenger RNA consists only of regions called exons that do code for the protein. The ribonucleic proteins in the diagram are small proteins that contain RNA. So, introns do not have information for proteins. Exons, they have information to make proteins. The ribonucleic nucleoproteins are going to eliminate the introns that are not needed for the production of proteins. Then, in editing, that will make different proteins, and that's why we have different proteins during editing. The protein will have more, or will be shorter. Depends on the protein. In the polyadenylation, we need a polyadenylation or a tail of poly-A. This is the signal that allows the messenger to go out of the nucleus to the cytoplasm, and that will help and protect the messenger RNA to not be linked to a different enzymes that may break it down. When we have messenger RNA that it is mature, it will go to the nucleus of the cell, and then the next process, that is translation, will happen. So